All right, for this next one, I'm going to do a problem that I gave a class in an exam a couple years ago. It's a little bit tricky, but you have the tools to solve it now. Okay, now I'm going to read the problem just to give it a biological context, but we're going to stick with some just basic letters. In rice, the mutations R, little r, okay, for round, okay, gap, G, and B, branched, okay, RGB. That's sort of a little in-joke there. Are all recessive alleles, so big R, big G, and big B would be the wild types, indicating the dominant alleles. Plants heterozygous for all three. You don't know what the linkage relationships were crossed. To plants homozygous recessive for all three, and the following phenotypes were observed among the offspring. And so what I mean by phenotypes is sort of the indication shown by the allele itself. So for the locus, you have the R locus, the G locus, and the B locus, okay? And I'm going to enumerate all the sort of implied genotypes based on the phenotypes here, okay? So there's big R, big G, big B, okay? Big R, big G, little b, and you can see what I'm doing here, big R, little g, big B, and then big R, little g, little b, okay? And now all the little r ones, little r, little r, little r, little r, okay? And then the same pattern, big G, little g, and then big B, little b, big B, little b. This is sort of a trick I just showed you actually for how to enumerate multiple sets. So you're sort of like, we'll do full alternation, big B, little b, so on and so forth. Then here you do GG, little g, big G, little g, and then you do the alternation there. That's sort of a way of enumerating all of those ones. And there should be 8 because there's 2 here, 2 here, and 2 here, possible alleles, so 2 to the 3 is 8. So this is a background there. So here are the numbers, okay? This is the number of offspring that were observed, okay? And the numbers were 23, 27, okay? 226, 227, 224, 223, 25, and 25, okay? And what's going on here, okay? Now, what we want to figure out is what these sort of phase relationships are of these individuals that are heterozygous. You know, are they all linked? Are they on different chromosomes? What's the genetic distance between them? And these data can actually make this figured out. So one way of doing this problem is to sort of break it down into pairs of loci, or pairs of genes. So you can say, well, what's the whether the R and G are linked, or whether G and B are linked, and you can also do whether R and B are linked. So let's just break it down first and say, are these linked, or are they independent? And if they are linked, what's the genetic distance between them? Okay. So in order to do that, we need to count up the different classes. Okay. So what are the sort of different classes? So with respect to R and G, okay, R and G, okay, there's going to be 4 classes, right? There's going to be, and I don't know if these are parentals yet, right? We don't know which is what, but we can actually go through here and just look at the relationship. So, okay, how many of this class do we have? Big R, big G. Okay, well, we have these ones here of that kind, and these ones here of that kind. Are there any others? Well, no. Okay. So there's 23 of those and 27 of those. So forgetting about B for now, we can say that there's 50 of this kind. Okay, now how about for little r and little g? Well, that's this set here and this set here. There's 25 and 25 for this 50 of this kind. Okay, that's kind of interesting. These numbers are not obviously chosen as a coincidence. And then so big R, little g, this class, and that's this class. Well, that's 226 plus 227, which I'll have to do the math there because I don't have it off the top of my head. 3, 1, 2, 5, and then 453. Okay, so 453 of this class. Okay, and little r, big G, 224 plus 2, so that's 447. 447. Now, first you want to ask yourself, okay, with respect to R and G, what's the parental genotype? Well, clearly these are the rarer kind, so these are the recombinants, and so now we can infer that this would be one parental genotype, and this would be one parental genotype, okay? So this is probably a bad representation up here, and let's fix it actually. Let's make it the way it is. So, and they're of course linked because it's not 1 to 1 to 1 to 1. Okay, so big R, little g, and then little r. Big G, okay, now how far apart are they? Well, these numbers probably look familiar. This would add up to 100, and this will add up to 900. Okay, so is there a recombination frequency of 0.10, or 10% recombination as a percentage, which would equal to 10 centimorgans. Okay, so we know now that these are 10 centimorgans apart. Okay, now let's ask a question about G and B, okay? Are they linked to each other? Okay, now clearly if G and B are linked to each other, okay, and R and G are linked to each other, then B and R would be linked to each other, okay? But let's just break it down and look at G and B right now, okay? So with regard to big G, little b, so what are the possible allelic combinations for G and B? Well, you can again, for G and B, we've got big G, big B, and little g, little b, big G, little b, and little g, big B. Okay, so what's the proportion of those? Okay, so let's count up the big G and big B's. Okay, so there's this one here, so 23. Okay, plus, where's another big G, big B? Okay, that is right here. Oh, 224. That's interesting. It's different, right? Because last time it was like small number plus small number, small number plus small number. Here it's like small number plus big number. Okay, well that's interesting. So what does that add up equal to? That's 247. Okay, so little g, little b. Okay, that would be this one right here, which let me erase these numbers because they're a bit confusing. Okay, so little g, little b is 227. Plus, where's the other little g, little b? 25. Okay. Okay, so that's 252. Okay, now you already know that we already calculated that we have 1,000 progeny, so something should be reminding you of something now. Okay, big G, little b. Okay, this one here, the 27 class. That's this one right here, big G, little b. And where's the other big G, little b? Oh, that right there, 223. Okay. Oh, that's interesting. So that is equal to 250. All right. Now you can actually add these ones up, or you can just subtract from 1,000 because you already know what the numbers all add up to be. Let's just do little g, big B. So that's this one here, 226, and then little g, big B. The other one's 25. So that's equal to 251. Okay. Now what's different about the first time? Well, with respect to G and B now, okay, there's a 1 to 1 to 1 to 1 segregation pattern. So what does that mean? It means they're unlinked, okay? Now, if you went and did this now, therefore, between B and R and B, you would see the same type of pattern of around 250 and around 250 and around 250 and around 250 for all the different 4 combinations. So what that means is, if we're to sort of draw the chromosomes here, you could kind of put these as a chromosome together like that, 10 centimorgans apart, semicolon, and then B on another chromosome segregating independently. In the last installment of these modules, we're going to do 3 linked genes.